Trying to figure out all my screens here. Oh. Okay, so I think we'll get going. Appreciate everybody joining us for the Michigan Wetlands Association luncheon here. We invited Tiffany to come talk to us about interdunal wetlands. Sorry, Dr. Schriever. But first of all, I wanted to say that the Michigan Wetlands Association, I'm on the training committee. We are trying to continue to do trainings. Part of having this webinar series is to keep engaged with our members, but we're still hopeful we'll be able to do in-person trainings soon. Right now, the board has decided not to offer in-person trainings currently. I'm hoping that'll change, and our goal is to start offering some trainings in August, and hopefully several at that point. We are looking to do part 2 of the wetland delineation training that we did virtually last month. For people who took part 1, they'll be able to attend part 2. We did have really good participation in that training, and probably won't be able to get everybody into the field portion, but we'll try our best to do that. We're also working with Tony Resnicek to offer a shrub course, and potentially James Lee to do a soil indicators course. We're looking at having winter twig identification in November, potentially. So if all this works out, we should have 4 or 5 really good classes for people to attend. So keep an eye out on that. We'll get emails out when the board decides to move forward with doing that. And keep an eye on the website. I've got the website on the screen. You can see that we've got our trainings on there. You guys who registered for this class should have registered through there. I think that's all the notes I have here. And so I'll turn it over to Dr. Schriever to introduce herself and talk about some of her research.
Okay, hi, I'm gonna share my screen here. Todd, it says I can't share because you are.
All right, try that.
Okay, this should work now. Okay, this one. All right, and if somebody could just let me know if they see the PowerPoint, okay.
Yep, we're good.
Okay, it's okay. I can no longer see, and I don't want to see myself anyway when I'm giving it. Okay, so thank you all for having me here today. I'm really excited to give a talk doing a luncheon series. I've actually come to a couple of your meetings before and hope to continue. So this is really fun. I admit I'm a little nervous, but very excited because I'm always excited about talking about interdunal wetlands. They're such a gem. And so it's good to share my enthusiasm and some of the neat things that I've found in them over the past several years that I've been studying them. So I'm going to give you a talk about the ecohydrology of interdunal wetlands along eastern coastline of Lake Michigan. And my work previously, well, and continually, has really focused on some aquatic systems that are less well known, less studied, not really given as much attention as those larger systems with lots of pretty fish in them. So I've studied temporary and small freshwater habitats around the United States, and these have been historically undervalued and really kind of understudied. Yet small and temporary freshwaters are, they dominate the aerial extent of continental waters. They take up more area than those large permanent systems. And they dot our landscape but they're often given very little protection and clean water regulation, as you know. So they're important systems for a lot of ecosystem functions. And I hope to gather more information about these small temporary and freshwater systems and more enthusiasm about them throughout my work. So I've looked at vernal pools here in Ontario, a lot of intermittent streams in the desert Southwest, and more recently looked at interdunal wetlands here on the Lake Michigan's coastline. Now, importantly, all of these freshwaters reside along this hydroperiod gradient from temporary to large permanent systems, and they experience really strong environmental variation. The temporalness of interdunal wetlands, of course, is on a different scale, it's on a different time frame than, say, vernal pools or intermittent streams, but still they can fall into that category that they do dry on occasion, right? So that environmental variation that all of these types of systems experience really dominates the I guess, regulation of community composition. And that's what I'm really interested in. So this talk will focus on interdunal wetlands. The first time I saw these, I was amazed that I moved here 5 years ago, and I just dug into them and decided to make my research program about them. So interdunal wetlands are wetlands that are found in low-lying areas within open dunes or between beach ridges along the Great Lakes. They do not have a surface water connection to a surrounding water body, so they could be considered isolated wetlands. They are naturally rare. They are formed in a couple of ways. One of the main way is when blowouts or wind forces the sand to move enough and open up a little bit of the bottom surface that connects with the groundwater surface. And so when that when the blowout is out and the surface water, the groundwater connects with the water table there, that's when these wetlands fill, and they can start percolating up water that way, and then precipitation is thought to then add to the rest of the water composition. As long as that water table is high, the wetlands should stay or should remain for as long as that water table is high. Now, there's a lot of different questions about how wetlands remain on the landscape in terms of their hydrology, and I'll be addressing some of that later on in this talk. But I just want to also clarify that these interdunal wetlands are different from other common coastal wetlands. They are a coastal wetland, but they're not the typical coastal wetland that you're used to seeing along Lake Michigan or at other Great Lakes. So these coastal wetlands, like I said, the interdunal wetlands don't have that surface water connection, and most coastal wetlands that people typically think of and that have been given a lot of study are directly tied to the lake, whether there's a barrier or not. They are do have direct surface water connection to the lake. And so Michigan actually has over 73% of the coastal wetlands that are found in the United States. So that's a lot. But a lot of them have been, sort of degraded or had some kind of disturbance to them. So we don't have as many as we used to, but they have been extensively studied and monitored for invertebrates, for vegetation, even for fish and amphibians. But there's a lack of information about these unique interdunal wetlands. So it's a whole set of coastal wetlands that have been basically almost ignored in terms of the information that we know about them. There's lots of good vegetation work along the dunes, but less so about the wetlands themselves. So that's where my interest lies, and that's where I really found that the information was lacking. And so I thought I would jump in and learn more about them myself. So they've been pretty vastly overlooked. They are embedded within the dune ecosystem. So where we find large concentration of freshwater sand dunes, which is here in Michigan, along the eastern coast of Lake Michigan, we also find interdunal wetlands. Ludington State Park has the highest number of interdunal wetlands in the world, which is a pretty cool fact to know and statistic to have. So even though we have this rare community type, we don't really know much about what lives in them, what's driving their composition, and even their density or the density on the landscape. So they have a unique ecological niche for animals and for plants. These animals and plants have to be adapted to fluctuating water tables. So like I said, they are sometimes here on the landscape, sometimes they're not. They do have rooted vegetation in some, while others are denuded of most or any kind of vegetation, as you can see from the picture on the top there. So they're all different in terms of vegetation and placement there in the dunes. So importantly, they do differ from other small wetlands, such as like vernal pools. They're very different in terms of organic matter content, how deep they are. They, of course, don't have as much of a subsidy in terms of influx of leaf litter or anything like vernal pools have. Their soils are very different. And of course, the canopy and possibly the animal communities are quite different. So initially, I just wanted to start learning more about what resides in these, and then what these organisms or how these organisms are responding to their environment. So I asked questions generally about that addressed how environmental variation influences ecological processes at multiple scales, from individual level to biodiversity patterns to ecosystem functions, both spatially and temporally. So, and that's because there's a need to understand the underlying mechanisms that maintain and also assemble biodiversity in order to predict how organisms will respond to changes in climate. Now, Michigan provides a really great, excellent study system to use natural gradients because we know that the pattern of species distribution and abundance will reflect the variation in an underlying environmental gradient. And this is an important driver of ecosystem structure and function. And Michigan, especially along Lake Michigan's coastline, it stretches over 4 degrees latitude, which creates a regional environmental gradient where we see a big change in floristic composition as we move from the southern to the northern ecotone. There's a decrease in the average precipitation and temperature from south to north, and potentially we will see there's a latitudinal diversity gradient going on here. It has been shown with species such as fish that there's a change in species diversity as you move along this latitudinal gradient in Michigan. But the latitudinal diversity gradient, it says that we would expect a relationship between species richness, and that species richness would increase with a decreasing latitude. And so we would expect to see higher species richness in the southern region of Michigan compared to the northern region of Michigan. So not only is Michigan interesting on this larger regional scale, because it's such a long environmental gradient, but it's also very interesting at the local scale. And when I say that, I mean at the local, like site level scale, we can see that as we move from the beach to inland, there is a gradient in environmental condition. And this is the dune succession gradient, which Michigan is well known for. The first ecological studies of succession were done here on Michigan dunes. So succession is a gradient of changing environmental conditions and constraints that affect vegetation. They influence the nutrients that are available. And these in turn influence the local communities that assemble. We also see there's a change from the beach all the way to the back forested dunes in terms of temperature, in terms of wind patterns, potentially the hydrology of these interdunal wetlands that are along this dune succession gradient changes as well. And this really, and as well as not just the environmental filters that are determining the community composition, when the community composition changes, the biotic interactions among those species also changes. And so these local communities are assembled through this environmental dune succession gradient and those species interactions. And so there are distinct plant zonations along this dune succession that is well studied and well known. What we know less about is the animals and how they respond, and the hydrology, how they respond along this dune succession gradient. And so my work looks at both this regional scale and this local scale to try to understand local and regional community patterns. So this, if you look at the pictures at the bottom, you see that early on, right after you pass over the foredune, that's when you start seeing interdunal wetlands. And those ones are out in the open dune landscape. There's no canopy cover. There's lots of light, lots of wind, kind of a harsh environment there. And as you keep moving along the different dunes, more vegetation is added with herbaceous grasses, some jack pine, some shrubs, and then you get into the back dunes where wetlands are more similar to, I would say, vernal pools, yet they're still considered interdunal wetlands in the more forested canopy area. So the coastal environment itself, at both scales, that regional scale with dune areas along the coastline, and at the local scale, so like within a dune area or within the lateral movement from beach to forested area along the coast, determine the abiotic and abiotic processes that would create unique freshwater biodiversity. So my study looks to try to look at that. We want to know what that biodiversity is, what is contributing to that biodiversity, and how biodiversity changes along these regional and local scales. So, so how does the environment shape community structure and function is an overriding question in my research. And so first we did some basic science to do some inventory of what the biodiversity looks like in interdunal wetlands. So we did inventory studies of amphibians, reptiles, and macroinvertebrates. I'm just going to tell you about the macroinvertebrate part of the study today. And then we started focusing on and examining the influences of that biodiversity. So what determines which community assemblages are where? So in terms of local and regional scales, and along those environmental gradients that we find across those scales. We looked at functional diversity as well to tell us a little bit about communities, the function of the community in terms of different biological traits. After we sort of got a little handle on some of the community assemblages, I've always been interested in hydrology, and so did a short dive into the composition of water with interdunal wetlands. And we looked at that in terms of what the interdunal water, the hydrology of interdunal wetlands along, of course, different gradients along the coastline, and whether there was any seasonal hydrological changes in interdunal wetlands. So I'm going to give you 3 little short case studies, hopefully not too long, to tell you what we found about these studies. So first, let's look locally. So a grad student of mine, Christopher Fraser, really started off my work in interdunal wetlands by looking at the spatial dynamics of macroinvertebrate assemblages along that dune succession gradient at Ludington State Park. So we asked whether those invertebrate communities and functional structure of those communities changes along that gradient. He sampled wetlands out in the open dune area here, where you can see we're very sandy, light colored areas, as well as wetlands in the forested part of the park. He did monthly sampling, so we had 11 different wetlands that he sampled every month from April to October. We took water chemistry at the same time, and he estimated the proportion of different aquatic plant types within each wetland, how much area they take up. And then we used GIS to calculate the terrestrial vegetation coverage within a 100-meter buffer of each of these wetlands to get an idea of not just the aquatic plant, but the terrestrial vegetation around them as well, to see what was driving some macroinvertebrate community patterns. So did we see any difference basically between those open dune wetlands and the more forested dune wetlands? So he collected and sorted and then identified over 42,000 individual macroinvertebrates from his study and found 102 different unique taxa, mainly at the general level. And what we found is that there are pretty big differences between open dunes and forested dune wetlands. They are very different in their community composition. The abundance in open canopy wetlands is dominated by midges and small-bodied organisms, and in the forested dune by larger predatory animals and different functional feeding groups. So we found there were 2 species that were exclusive to open dunes and were not found in forested dunes. Those were some midges and microcaddis species. And in the forested dune, there was 13 different species that were only found in the forested dune habitat. Notably, those had different functional feeding. They were of different functional feeding groups than what were found in the open dune. So the Limnophilus caddisfly was not found in open dune wetlands, mainly because there's really not any leaf litter for them to shred. Really, a couple of different genera of Dytiscid larvae, so bigger predaceous diving beetles. And importantly, if you want to avoid mosquitoes, just avoid the forested area, right? Mosquitoes were only found in the forested dune wetlands and not ever found in the open dune wetlands. So they're compositionally unique and functionally unique as well in terms of the functional feeding groups that were found in each of them. If we look at whether these compositional differences are due to different environmental factors or just basically where they're located in space within the dunes, we also see differences. So if we look at this, let me break this figure down for you a little bit. On the y-axis, we have Bray-Curtis dissimilarity, which higher numbers mean that your community composition is more different from another. So these are pairwise comparisons. So one wetland to another wetland, how are their community compositions different? If you have high dissimilarity, your communities are more dissimilar. And then I have the difference in terrestrial vegetation on the x-axis. So if the terrestrial vegetation is very similar to one another from one pond to another, you're over here on the left side, and if there's a big difference between 2 ponds with vegetation types you're going to be over on the right side. And so if you compare an open dune to a forested dune, the difference in the terrestrial vegetation is very high. And basically 0 versus, 100% of difference. And then also these ponds are located in space. And so if wetlands are near to each other versus wetlands that are farther apart from each other are noted in the color gradation scale here. Farther apart have darker blue colors. So what this figure shows is that wetlands that are more similar in community composition, so these lower dissimilarity values down here, tended to be located closer together. So the lighter blue physical separation coloration, and they have less difference in terrestrial vegetation cover. So if you compare two open dune wetlands that are near to each other, they are very similar in species composition. But if you compare an open dune to a forested dune wetland that are farther apart from each other, they're going to have very different species compositions. So the open forested had more dissimilar, so the open versus forested had more dissimilar communities. And also, as wetlands became more physically separated in space, their corresponding invertebrate communities also became more dissimilar. But that terrestrial vegetation was driving a lot of the differences in species composition. We can also see this here in terms of how taxonomic diversity varies along the dune succession gradient. Shannon diversity here, if Shannon diversity was highest in areas that were dominated by a higher vegetation cover, by more of a neutral pH in the water, low dissolved oxygen, and those wetlands that were farther inland. So, so diversity was actually higher in the closed canopy wetlands. The shallow open dune wetlands, they may have more algae, but that has influence on water chemistry. But forested dune wetlands have a really big buildup of organic matter, which can change the pH, as well as if that organic matter is all decaying, it uses more oxygen in decomposition. So those wetlands tended to have lower dissolved oxygen than the other.
So Tiffany, we got a question that
Just saw that.
Curious if sandfly populations might be higher in areas with ICW. Well, I'm not sure what sandflies are.
I don't either.
Okay, all right.
What's ICW? Well,
Intercoastal wetlands, interdunal wetlands, maybe.
okay.
Is my guess. So no mosquitoes in the open wetlands.
No mosquitoes in the open wetlands. There are midges, right? They're all non-biting midges mostly.
All right, I'll let you know if we get more questions.
Yes, thanks. It's hard for me to monitor that. Okay. Also, let me know if I'm going too fast if you want me to.
No, it's fascinating. Very good. And yeah, I'll keep an eye on chat. I know it's really hard when you're presenting, so don't worry about that at all.
Thank you. Okay. So yeah, so the environmental gradient along the dune succession gradient was driven by these different aquatic, different water chemistry measures. And oops, there we go. Okay. And so we found an increase in species richness as well as functional richness with the distance from the lake shore that corresponds to those underlying environmental environmental factors. So functional richness might be a newer term for some, but basically it measures the amount of niche space for that a community takes up. And so with larger functional richness, you have a larger realized niche space and greater combinations of functional traits. And so that makes a lot of sense that we saw that there was more functional feeding groups in the forested dunes than in the open dunes. So there's just a lot more ecological niches available, and they're taken up with different trait combinations that aren't found in the open dune wetlands. So it's not bad to have a lower functional richness, it's just different, right? And so in general, open dune wetlands tended to have lower functional and taxonomic richness. And this is the functional richness pattern as well as the species richness pattern is what I can find one of the first recognized pattern for aquatic organisms across the dune succession gradient. We've seen differences and patterns of this in terms of terrestrial species and terrestrial vegetation, but I haven't been able to see results like this for aquatic communities. Okay. So if we move on to the regional scale patterns of macroinverted assemblages, this was spearheaded with Michigan Sea Grant money and Nicole Stewart, a previous graduate student work. Could not have done it without her help and her dedication to both field work and analyzing a very large number of macroinvertebrate samples. So this is a lot of her master's research here. So we asked the question about whether aquatic macroinvertebrate diversity and composition varies along the coastline. And we had predicted that there would be high species replacement, which would be correlated to that environmental gradient, both locally and within a park and regionally along the coastline. And we thought we would see that increasing species diversity with decreasing latitude. So how we went about testing this is that we sampled locations, 5 different parks along that north-south gradient on the eastern shore of Lake Michigan. We sampled Indiana Dunes, Warren Dunes, Saugatuck Harbor Natural Area, Ludington State Park, and Sleeping Bear Dunes. In total, we had 38 different wetlands that we could end up using for a total of 249 collections of macroinvertebrates. Today, I'll be showing you data from the collections from April, June, August, and October of 2018. Okay. So at the same time, we measured local, so within wetland conditions to correlate with the biodiversity metrics, and as well as we took regional factors that we were able to get from WorldClimb website to look at the environmental conditions along the latitudinal gradient. We use these environmental factors to understand the underlying gradient driving species diversity patterns. And then we quantify invertebrate diversity at a couple of different levels. So we can quantify invertebrate biodiversity at the pond level. So that's alpha diversity, that's the species richness within each wetland. We also can look at the total richness of all the wetlands that we sampled along the coastline to represent our gamma diversity or our regional species pool. So how many species are actually available to be part of each of those wetlands? And the change in species composition among those wetlands is our beta diversity. So we want to know the change between wetlands and the change between regional species, between the regional species pool. And the concept of beta diversity is important because it connects biodiversity at the local scale, so at alpha diversity, and at that regional species pool at that gamma diversity. And so a higher beta diversity, again, illustrates there's lots of change in species composition from one wetland to the next or from one area to another. And then we can partition beta diversity into a couple of different components, into a subset and a replacement component to determine the underlying factor that controls that beta diversity. So we have subset beta diversity, which presents itself as a nestedness pattern, where you see that the species in one community is just a subset of the species of the overall community in another one. So if there's 4 different species in one wetland, you move to another wetland and there's 3 of those same species, but not the other. That's a subset of that wetland. The other partition of beta diversity is called replacement beta diversity, and this is when you see a turnover in species composition from one wetland to another across an environmental gradient. So for instance, the species richness might be the same between, say, wetland 2 and wetland 3, but the species identity is different, and that's represented by that turnover. And so beta diversity is our measure, and beta diversity is measured as community dissimilarity. So the more dissimilar a community is from one another. We use Mantel tests to test if community dissimilarity is correlated with factors at different scales. So at the local, regional, or whether just geographic distance has anything to do with our diversity patterns. So we tested each of these separately. So the beta partitions, the total beta diversity and the replacement and the subset were tested separately for at each of those local, regional, and in terms of geographic distance scale. So that might be slightly confusing, but when I show you a figure, you'll understand. And this is to look at species richness and diversity patterns across different scales and driven by those different factors, and whether we can partition out what factors are driving that compositional change. So some results for you. So we had a really wide range of species richness among wetlands and among different parks. So it ranged from 58 different species to up to 83 different species within each park. Overall, there was 110 different species. This is I'm saying species richness, but it's really general richness. And so the pattern is that there was higher diversity overall in the southern ecoregion. So Saugatuck Harbor, Warren Dunes, and Indiana Dunes, Lakeshore overall had higher diversity and richness than Ludington and Sleeping Bear. And there was a significant relationship with species richness and latitude, in that those lower latitudes had higher species richness than the higher latitudes. So we did find that latitudinal diversity gradient relationship. Now, what is that driven by, right? So it's not just the latitude that's driving the change in diversity, but it's the underlying conditions that we see that change along that latitude that are driving diversity. So what is influential along the latitude that could be driving this pattern? Temperature and precipitation are related to latitude, but we're not related to diversity change. The only environmental variable that we found that was related both to latitude and to species richness was pH. So pH increased along the latitude, and so higher latitudes had higher more neutral basic pH, and diversity declined, of course, and richness declined as the latitude increased and pH increased. I can make some assumptions that pH, changes with changes in vegetation communities and soil communities, but I don't exactly know if there's a stated relationship out there of how pH changes along Lake Michigan's coastline. I'm not sure. This is something that was a surprise to us and interesting that we need to follow up on. In terms of community composition, though, this is where we see the most interesting results. We see that in terms of beta diversity, so dissimilarity among those aquatic macroinvertebrate communities, we see that we see moderate and similar amounts of beta diversity change across dune or among dune areas, ecoregions, and for the entire coastline. So there's overall fairly high beta diversity change. So if we look at the y-axis is beta diversity, and then each of these plots shows the different factors that underlie that diversity change. So geographic distance, so sites that are farther apart from each other generally had higher and moderate levels of dissimilarity. The regional scale environmental factors also showed that when there's higher differences in regional environmental factors, differences, then you have higher beta diversity change, and also at the local environmental patterns as well. And then those three different levels of beta diversity, total beta diversity, so the total change in community composition between wetlands, the replacement, which is the turnover in species, also contributed to that change in species in community composition patterns. The total dissimilarity so the total beta diversity and the replacement beta diversity were both driving changes in community composition in terms of geographic space and along the regional scale, but then the subset beta diversity was also important at the local environmental scales, but wasn't at the other scales. And so interdunal wetland macroinvertebrate communities were moderately to highly dissimilar, despite being located within this shared dune landscape. And so this indicates that both environmental factors as well as space, so geographical location, drive aquatic macroinvertebrate beta diversity patterns. So these numbers here I'm showing is the beta diversity values for each park. So higher values indicate more dissimilar communities as you move from wetland to wetland within each of those parks. So what we see at that there's moderate to high compositional dissimilarity within each park, which was dominated by that turnover, that replacement of species from wetland to wetland. Within each dune area, they all have very similar values, and that this is repeated along each dune area and within any of those regions. So the regions in the northern region and the southern region both had very similar beta diversity values, dissimilarity values. And this is shown across the entire coastline as well. So this is a unique pattern that is not seen a lot, where the beta diversity and the replacement are repeated along that environmental gradient. So we know that environmental gradient is probably driving this change in community composition. If we run a test to remove that environmental fact, does just space itself explain to us about this beta diversity patterns? And after we do that, we find that spatial control is very limited, actually. And so this means that interdunal wetlands were not limited by dispersal, that organisms can move around this coastline, creating changes in species composition among each of these wetlands and these dune areas. The environment, so this indicates that environment is driving this community structure along this coastline and within each of these interdunal wetlands within these parks. The dune succession and the regional environmental gradient were really important then, and we know little about organisms that live. So now we know a little bit more about the organisms that live in these wetlands, and this represents a little bit on the first comprehensive study of aquatic communities in interdunal wetlands and what sort of drives their composition. So it looks like it's already 12:45. I've until 1, right?
Yeah. Yep.
Okay. Okay, so I'm going a little slower. I also wanted to show you a little bit about the hydrology work that I've been doing. And so I was still thinking about water, right? And we have these very different open dune systems versus forested dune systems. They seem to dry down and fill up at different times or rates that I saw. So we know that these wetlands are connected underground through Lake Michigan. We know that they form because the water intersects with the water table. And through some of the research earlier, I can see that the water level, the depth of the water in interdunal wetlands kind of mirrors the pattern of the water level change in Lake Michigan over a particular season, where we have rising water levels early in the season with a height sort of around July and August, and then water levels come down again. And so that assumption says that then interdunal wetland water is probably highly driven by lake water levels. And they exhibit yearly as well as seasonal fluctuations. But what is actually, is the water 100% Lake Michigan? Is it driven just by Lake Michigan, or it can be driven by a whole bunch of other water sources? And so my objective was to characterize the water sources of interdunal wetlands and to use stable isotopes to do that, because different water sources have a different isotopic composition. And so we hypothesized that the wetland hydrologic connectivity of Lake Michigan would deteriorate with distance from the lake and could show some seasonal patterns of when Lake Michigan's water is more important for interdunal wetland water and compared to others. So I took a smattering of 6 different wetlands within 4 different parks in Michigan, in the northern latitude or the northern ecoregion and the southern ecoregion. And so in total, we had 24 different wetlands that we were sampling for core water, so a shallow groundwater. We sampled in June and August and October of 2020, and took those water samples and analyzed them for stable isotope oxygen and stable isotope hydrogen, so the isotopes of water. And so we also took potential water sources that can contribute to the water within these wetlands along the area. So we took 14 different surface water samples that were from precipitation, Lake Hamlin, the Kalamazoo River, which is nearest to the Saugatuck Harbor Natural Area, Lake Michigan water samples, and we also took Platte River water samples. So these different sources could be contributing to the water balance of interdunal wetlands as well. And we ran those for stable isotopes to see where they fell as well. At the same time, I collected macroinvertebrate samples because, of course, I have to whenever I'm in a wetland. Unfortunately, I can't give you information about these ones yet. I haven't finished IDing those all yet. So what did we find? So in terms of an isotopic relationship, we look at the relationship between oxygen and hydrogen, and we can see where our water samples fall in relationship to the local meteoric water line as well as the global meteoric water line. So all those blue dots are all the water samples that I took. And the 2 dashed lines represent the global and the local mean water line. And so if you look at this, you see the solid line is our regression fit of all of our water samples. And we see that the slope of our regression fit is a 5, which is less than 8 and has an intercept less than 10, which is your global standard of precipitation. So our wetland samples basically fell above the local mean water line, but had a lower slope. So this tells us that we do not see any, so we do see evaporation in water sources at each of these wetlands, but we have a very wide range in oxygen and hydrogen from each of those samples. So there's really wide range along the coastline of isotopic composition, but there's no seasonal difference in those compositions. And this shows that precipitation is not a major source of water to these wetlands. If we look at this relationship, to compare the relationship of oxygen to hydrogen for each park, we see that each of them have significant and positive relationships, and the relationship is similar across park, but the slope of each of those wetlands is different, and each of the slopes is still less than the local meteoric water line. But this is different in that each park is showing a slightly different relationship. Compositionally, the waters in the wetlands within each park are significantly different from each other, which may be different from the water in Lake Michigan. So we can see that, but it does not seem to have a latitudinal relationship. So Ludington, Sleeping Bear Dunes, Saugatuck and Orange here, and then the green is Warren Dunes State Park. Compositionally, the hydrogen isotope is different among each of those, except for Saugatuck and Warren Dunes, which had similar hydrogen isotopic composition. Oxygen was kind of all over the board. They were all sort of different from each other. But so this tells us what the isotopic composition of the wetlands was, but I wanted to really know whether each wetland had different water sources. So if we look at each park individually, we can see the variation in water source. So now I have hydrogen on the X, or on the Y, excuse me, oxygen on the Y, and then the blue dots are the interdunal wetland water, and the black dots represent the different sources for each park. So I have Lake Michigan, Lake Hamlin, and precipitation for Ludington State Park. Sleeping Bear Dunes has the Platte River water source. And from each of those, we notice that there's a wide range, right? The wetlands are kind of all over the board. So some of them might be solely Lake Michigan water. Some of them might be solely Lake Hamlin water. Others are just a mixture. And they don't seem to be, and so they seem to be a mix of both Lake Michigan and Lake Hamlin. For precipitation, again, does not seem to be a very big source in either of those water, in the makeups of any of those wetlands. And in Ludington State Park, we see that some of them there is a trend for the distance to the lake to influence the hydrology of each of those wetlands. The ones that are farther away are different isotopically than Lake Michigan water, and the ones that are closer. Same for Sleeping Bear Dunes. The speed over here. And then we have Warren Dunes State Park and Saugatuck Harbor Natural Area, and what their interdunal wetland water composition looks like. If we look at Warren Dunes State Park, really don't know what the water composition is coming from there in terms of what we found. There's not another water source nearby. The closest is the St. Joe River, which is 20 some miles away. So, and the water sources probably are not coming from that here. The Saugatuck Harbor Natural Area is interesting because a lot of these wetlands fall really close to Lake Michigan and really close to Kalamazoo River water. But there are a few that are very different. So this is from 2 wetlands that were sampled in October. The water composition is a mix and is different. This one way down here at the bottom is closest to precipitation. That is a wetland that just formed 2 years ago. And so that makes sense that its water composition is more precipitation driven, given that it's so new. So we see a reflection of different water sources for different wetlands. And I'm going to probably end, I think, because it's getting late if you want to ask any questions. But we also looked at the composition of water in terms of water chemistry too. And I have data on that if you want to see that later. But at that, I guess I'll just end by saying that we did see very separation of water sources. So we are able to tell a little bit about interdunal water source is variable. It doesn't happen to be seasonally. It may have a trend to be distance from the lake, but the variation or the variance on precipitation varies across wetlands and doesn't seem to be a large importance. Lake Michigan is a contributor, but not the dominant contributor, what we found in terms of the hydrology of interdunal. I say thank you to all my wonderful graduate students that participated in this work, and of course my funding sources. And thank you for having me on today.
Yeah, thank you, Dr. Schriever. Really interesting, good stuff. Keto had a question about, any thoughts on why there are not mosquitoes in the open wetlands?
Well, it's probably hard for them to oviposit on the water, given there's so much wind right there. Especially in Ludington, it's quite a windy area. Mosquitoes don't fly well in a lot of wind. But I mean, food-wise, they would have the ability to filter through the water and get algae and fine particles. So I'm not entirely sure why, but we've never found a single mosquito larvae in interdunal wetlands.
Fascinating.
Yeah.
So what we love to categorize everything and put everything in groups. And what I'm hearing you say is that would be extremely difficult. Each of these wetlands appears to be slightly different.
They're different.
You're gonna figure that out?
Yeah, no, I mean, you can. So we've done like some analyses to look at that, and there seems to be like open dunes and some like intermediate level different ones that have a little bit more terrestrial vegetation and even some some more herbaceous plants that are growing in them, and then the ones that are forested. So you can group some of them that way, and that does work, but compositionally, in terms of their macroinvertebrates, it's easy to categorize forest versus open dune. Those are very, yeah.
Can you see the chat there? We got Glenn is asking, could vertebrates, frogs, or small fish account for some differences, such as lack of mosquitoes?
Possibly. There are no fish in any of these wetlands, so that's not the reason. We do have some frogs that of course breed in the interdunal wetlands, mostly toads. You see a lot of green frogs actually. There are salamanders in some of the forested wetlands that we sampled, so that could be some of the difference. I know salamanders love to eat mosquitoes, but there are no fish to drive any of the differences here.
Okay, well, I think that's our time. We really appreciate your time. Really interesting, and we'll like to hear an update in about 5 years when you figure it all out.
Yeah, sure. If I figure it all out.
Oh, I have no doubt.
Yeah.
So thank you everybody for joining us. We're going to try to schedule another one of these for next month. So if you got any ideas, or if you want to do one yourself, get a hold of me or Amelia. Amelia sent out the emails, I think, to everybody. So thank you again. And thanks, Dr. Schriever.
You're welcome. Thanks for having me.
Thank you.
Have a good day. Hey, Todd, are you staying on for a minute?
Yeah, I can.
I don't think Nicole got to come today.
She was on there. She did.
Was she?
Yep, she did chat. Yep.
Probably murdered her section of the, of the presentation.
Yeah, I think she is off. So, but yeah, I chatted when you gave her a little shout out. I chatted to her. So yep, she was listening.
Yeah, she was really good. She was pretty busy. Well, I went too fast and didn't explain things as well as I wanted, but hopefully they at least get that. They're pretty darn cool wetlands, and we don't know a lot about them. So if anybody else wants to jump in and yeah, try to learn more about maybe the soils would be interesting.
Oh yeah, yep. Well, and they are, I think, just on a broad level on identifying hydric soils, and we may have talked about this before, but they were not recognized as even being wetland soils by the National Council on Hydric Soils.
Mm.
And so after they published, boy, it was they put out, when they did the wetland delineation manuals update in the early like 2010 timeframe, when they put
Mhm.
those first manuals out on how to identify hydric soils, we found large areas in Michigan, mostly on the coast, that were not identified as hydric soils. So they did pretty substantial study in those areas and found that they were hydric soils, and then they're now included. But I mean, it
Good.
was even at that level, just the identification level, no one really had done much work in those areas because the soil scientists have always focused on agriculture. There's no
Mm.
agriculture, so
Mm.
they kind of missed that. Um, so no, I thought it was great. I'm a little confused on the beta diversity thing, so maybe we could talk about that sometime.
Yeah, I should have spent more time trying to explain it. Yeah.
Um, well, yeah, it's actually
Actually,
not.
I was afraid I was going to lose everyone, but it's really, yeah, I should have done a better job. I'm afraid of that because there's so much information in there that I explain in that way.
Yeah, yeah, yeah. I was, and I didn't, maybe people got it. I was also trying to. We had some issues with our link, so I think we lost a few people, but
Oh, bummer.
yeah, it was a little strange that the email that Zoom sent out had a Google link, and it actually didn't go to the right place.
Oh,
So it had the Zoom link too. So if they had gone back and clicked the Zoom link, they would have got in. But and I think we probably got most of the people who wanted to attend, but it took a little bit. I was dealing with that,
all right.
but
Gotcha.
it's all good. No,
Okay.
good. It's fascinating. It's really interesting. You found a pretty good niche there, I think.
Yeah, I think so.
Um,
I just wanted